In this part, I'm going to show how to run a similar analysis that I've already described for the whole UMAP, but this time in a single cluster. And this way we'll be able to learn more about aging. So I'm going to click now on the second tab called Cluster UMAP. So let's look into the gene col2 that we've already used in our first part. And remember, this gene was expressed in cluster number 10, the VD motor neurons. So I'm going to look particularly in this cluster. I pick cluster number 10. I also decide to color the cells by gene denoise. And I'm going to look at the gene col2. I click show results. And the analysis results in these 5 panels. The top 2 panels represent the UMAP of the whole data set, which pretty much summarize what we found here. The left UMAP shows you just the position of this cluster, and the right UMAP here color the cells by expression of col2 in the entire gene set. So remember, they are in the right cluster, cluster 10. Now the lower 2 UMAP here are a new projection of the cells coming only from this cluster, the one that we've picked. This on the left UMAP here, you see the cells colored by age, and on the right UMAP, you see the cells colored by expression of col2. You see that the cells seems to be enriched, the expression seems to be enriched particularly at certain age. So we can also use the third panel here, which is a graph that shows every cells of this cluster and in function of the expression of col2 and their age. And we can see that indeed the expression of col2 goes up past day 5, at day 8, and particularly probably at day 11, which we only have 2 cells. And from here you can run the same type of analysis I have described earlier for geneset. So you can pick, instead of gene denoise, say AUCL pre-computed, and access all the transcription factor target genesets, and then run them in any cluster of your choice, like that. And the same thing applies to it will run. Yep. The same thing applies to the anatomy marker genesets or the custom genesets.